Is genetic modification making our world better or is not? We think we know our planet, but still a secret world to be discovered. Genetic modification is the process of changing or altering DNA, the genetic material of an organism. This can involve the insertion, deletion, or the modification of specific genes. This technique has, for example, been used to develop fungi and bacteria that produce medicine. Modifications of microorganisms is mainly used in pharmaceutical applications. In plants, we make the distinction between cisgenesis and transgenesis. Within genetic modification, cisgenesis is the transfer of genetic material derived from a related plant. For example, Wageningen University and research has utilized this method to use genes from wild potato plants to make consumer potatoes more resilient to phytophthora. Transgenesis is the transfer of genetic material from non-related species. Genetically modified organisms are plants or animals with certain genes altered in their genetic material. In other words, genetically modified organisms are organisms which include microbes, plants, and animals, whose DNA has been modified using some genetic engineering techniques. The National Academy of Science, the United States' #1 source for independent, objective advice to the nation on matters of science and technology, conclude that GMO are safe for human health. But is that true? Are the research labs making things beyond what we know? We're the furthest we have come in the history of genetic modification to date. Reproduction of human DNA. Replication is the process by which the genome DNA is copied in cells. Before a cell divides, it must first copy or replicate its entire genome, so that each resulting daughter cell ends up with its own complete genome. Clonation of insulin gene. In 1973, biochemists Herbert Boyer and Stanley Cohen developed genetic engineering by inserting DNA from one bacteria into another. In 1982, FDA approved the first consumer GMO product developed through genetic engineering, human insulin to treat diabetes. Genetic investigation to develop vaccines. All vaccines are genetically modified in a way. A gene may be programmed to produce an antiviral protein in a bacterial cell. Once sealed into the DNA, the bacteria is now effectively reprogrammed to replicate this new antiviral protein. Development of transgenic food. In a lab, the genetic makeup of a plant is changed, typically by adding one or more genes from its genome, to create genetically modified plants. The new transgenic DNA is intended for the plant cell's nucleus. The clonation of animal with success. Dolly was a female fin Dorset sheep and the first mammal that was cloned from an adult somatic cell. She was cloned by associates of the Roslin Institute in Scotland, using the process of nuclear transfer from a cell taken from a mammary gland. Her cloning proved that a cloned organism could be produced from a mature cell from a specific body part. Contrary to popular belief, she was not the first animal to be cloned. After cloning was successfully demonstrated through the production of Dolly, many other large mammals were cloned, including pig, deer, horses, and bulls. The attempt to clone Argali, mountain sheep, did not produce viable embryos. The attempt to clone a Bantam bull was more successful, as were the attempts to clone Mouflon, a form of wild sheep, both resulting in a viable offspring. The reprogramming process that cells need to go through during the cloning is not perfect, and embryos produced by nuclear transfer, often of normal development, making cloned mammals was highly inefficient. In 1996, Dolly was the only lamb that survived the adulthood from 277 attempts. By 2014, Chinese scientists were reported to have a 70 and 80% success rate cloning pigs. And in 2016, a Korean company, Soan Biotech, was producing 500 clone embryos a day. Wilmut, who led the team that created Dolly, announced in 2007 that the nuclear transfer technique may never be sufficiently efficient for use in humans. Genetic modification banned. Reasons to ban germline gene editing include the profound risk to future children, thin medical justifications, reinforcing existing inequalities, and creation new forms of discrimination, eroding public trust in responsible science, and undermining global agreements. Why it is an issue? Environmental concerns include the risk of outcrossing, where genes from GMO foods pass into wild plants and other crops, a negative impact on insects and other species, reduction in other plant types leading to a loss of biodiversity. What is the future of genetic engineering? With genetic engineering, it would be possible to study gene functions and come up with vaccines, life-saving drugs, and many other things like the reduced need for pesticides. We may even find a cure for cancer. In conclusion, if GMO was used in a good way, it would cure hunger in developing countries like our country. Also, curing viruses like nowadays we are facing as COVID-19. Making a global change in health and quality of life and build a world that supplies food and well-being accessible for everyone in that way that GMO could be considerable as good for us. This teaches us that we have a lot of things to learn, but even more to discover.